Hello and good evening, everyone. Welcome to night number 3 of our summer research experience student presentations. My name is David Dimitri, and I'm the program director at Headwater Science Institute. And in case you're not able to join us the last 2 nights, we've had over 20 students take part in our research program this summer. And tonight we welcome the final group of students who will share all of their hard work with you. Now, once again, tonight you are also going to have the opportunity to meet some of their incredible mentors who have been helping them out along the way. Now, before we jump into our presentations for tonight, all right, so let's get into tonight's presentations. For those of you who are watching live on YouTube or Facebook Live, we encourage you to share any questions or comments you have for our researchers throughout their talk. After each talk, the researcher will have a few minutes to answer any questions from our audience, so please be sure to add any comments or questions along the way for each of our students. So now I'm very excited to welcome our first mentor. Hi, Katie.
Hi. Happy to be
Katie's
here.
going to go ahead and introduce our first researcher for tonight. So go ahead, Katie, and take it away.
Well, I had the extreme pleasure of mentoring Annette this summer. And Annette is a high school senior in Washington State. And when we first met, she expressed an interest in fungi, which was new for me because I have a background in plants and insects, but not fungi. But I've always been fascinated by them. So I was really excited that she had this interest. And because of that, I actually learned a ton about fungi. It's a really exciting area. And she settled on a project where we actually took data which was previously published. So, but beyond science, Annette has a lot of interests. She's very humble. And so it took a while for me to get some of this out of her, but she is an accomplished ballerina, and she's been dancing for over 10 years. And in addition to dancing, she loves to hike, and she recently took a trip to Mount Rainier with her family. And yeah, it's been great getting to know Annette and learning more about her research interests, and I hope she continues learning more about fungi in the future.
All right, thank you, Katie. All right. All right. So for my summer research, I studied the latitudinal gradient of fungal genetic diversity with my mentor, Katie. So firstly, what is a latitudinal diversity gradient? Well, a latitudinal diversity gradient is a pattern of species diversity that increases near the equator. So as you can see in this image map,
Annette, I'm going to stop you for a second. I'm sorry, we're not seeing your presentation on the...
Oh, is it not working?
Yeah, so go ahead. Exactly. We'll stop sharing for a second.
All right. Should I try again?
Yeah, go ahead and try the share again along the bottom.
Okay. Is it on now?
All right, so we have it on the screen. Go ahead and click on present, and we'll make sure it gets started for you this time.
All right. Can you see it now?
Yes, we can see it. So go ahead and take it from the top again if you don't mind. You can start over.
Right. Sounds good. So once again, this summer I researched the latitudinal gradient of fungal genetic diversity with my mentor, Katie. So firstly, what exactly is a latitudinal diversity gradient? Well, it's a pattern where species diversity increases towards the equator. So as you can see in this map below, The warmer colors represent areas where species diversity is higher, and the cooler colors represent areas where species diversity is lower, which are further from the equator. They've been observed across many taxa and kingdoms, such as animals and plants, and this particular map shows it for living terrestrial vertebrates. A lot of hypotheses have attempted to explain this strong pattern, and for this particular project, I decided to research fungi extracted from lichen, which are symbiotic organisms composed of fungi and algae. Previous studies suggest that this latitudinal diversity gradient applies to fungi, so I based my hypothesis on this fact and assumed that it would apply to lichen as well. So my research question was, does the latitudinal diversity gradient apply to fungi and lichen? My independent variable was absolute latitude, which simply told us the distance from the equator of samples that were taken. And my dependent variable was the total number of ITS variants. Now, ITS stands for internal transcribed spacers, which is a region on the genome of numerous organisms, in this case fungi, that can be used to identify, that can be used as an indicator of species diversity because it's highly conserved within species and varies between species. So for my hypothesis, based on previous studies suggesting that the latitudinal diversity gradient applies to fungi, it likely applies to fungi extracted from lichen as well. So my materials and methods. My main program that I used was the Global Fungi Database, which simply collected all of the data from published papers on fungi samples. And what I did was I first went to the site and used their GeoSelector, selected the entire map of the world, and downloaded data for all available sites. I then sorted by lichen as my source, and I combined observations that were conducted at the same site just to remove any repetition. And based on my independent and dependent variables, I chose a linear regression, which would best show the relationship between the two. So for my results, as expected, the relationship was negative, which meant that the further you moved from the equator, the lower the species diversity would be. My p-value suggested that my results were significant, and my r-squared value was relatively low for reasons that I will get into on the next slide. So again, evidence suggests the existence of a latitudinal gradient in lichen fungi diversity, thanks to my p-value that I found. But the low explained variance is possibly caused by variation in sampling methods across studies, because since I was analyzing pre-existing data, I didn't have the opportunity to go out and collect it myself, which meant that people could have used different methods of collecting the samples, which could have caused some variation. And from this, we can conclude that latitude is an important predictor of genetic diversity in lichens. So for future research, I believe that I would like to have more research done in more tropical regions, because in the graph shown here, the lowest latitude that is provided for this particular sample is around 40 or 50 degrees, which is not ideal given that it would likely be highest towards the equator. And more controlled data collection would also be better, because this was most likely one of the greatest sources of error in my research. And fungal diversity based on elevation would also be a good field to go into, because while I was doing research, I found that lichen diversity varied greatly based on elevation above sea level. And so I thought that given that there's a lack of large amounts of data on lichen, in fungi, I decided that would likely be a good idea to pursue this as well. Oh, sorry. These are my works cited. Does anyone have any questions?
Great job, Annette.
Thank you. Well, one of the main challenges that I had working with this type of data was really the analysis, because prior to this research, I had not really done much data analysis, but my mentor Katie helped me a lot with that. And one of the biggest challenges I faced was just the sheer volume, because we were dealing with thousands and thousands of rows initially, and so we had to cut out some and take random samples. Yes, I'd like to pursue more of this research in the future. I think it's really a field that I'm interested in, because fungi are all around us, but they're often overlooked.
All right, and excellent. Thank you so much for sharing your research. Yes, excellent work, and really cool stuff to hear about kind of a global data set like that, and really interesting to see kind of where you might go with it next. So thank you so much for sharing. And Katie, thank you so much for helping Annette along the way. What we're going to do next is actually keep Katie on, and she is going to go ahead and introduce our next researcher for tonight.
Awesome. Okay, so my next student, it was also quite a pleasure to be able to work with her. Farida is starting her sophomore year of high school in the Bay Area, and she's going to get involved with lots of clubs this year, including Green Team, which is an environmental club, Mock Trial, and Model UN. Farida lived in Egypt until she was 10 and is actually trilingual. So she speaks Arabic, French, and English, and wants to learn Japanese. So very impressive. Over the summer, we usually met at 10 a.m. on Mondays, which is a little bit of a struggle for me, but Farida was just full of ideas and passion and energy. And so it really helped me get my day started and really my week. So I really appreciate that from her. And she came to me with a lot of thought-provoking questions, but ultimately we settled on a field experiment that she conducted in a nearby nature reserve. And any good field biologist can tell you all of the trials and tribulations they encountered on the field, and Farida is no different. She encountered not one, but 2 coyotes while she was doing field work. So if she can stick with it through the coyotes and the mosquitoes and everything else, she's going to go on to make an amazing field biologist. So thank you, Farida, and excited to hear what you have for your presentation.
Great. Are you able to see?
So we're not able to see your screen yet, Farida. If you could go ahead and try that share button again at the bottom of your screen.
Okay.
And you should be able to pull up your slides on the web browser again.
It says it's sharing.
Okay, let's see. There they come. All right. So we'll go ahead and you can go ahead and get started. Thank you.
Okay. So I researched, or I looked at, the distribution of native and non-native plants in relation to hiking trails. Some background information. Firstly, what are invasive species? So there's plant species that are not native to the region. They're usually introduced accidentally or purposely by humans, or possibly even by animals or some other methods. how do invasive species become so successful in regions where they're not native? They're not supposed to be there. So there are a lot of ways they can be successful. Mainly, they either produce large quantities of seeds or they have far-reaching deep roots, or they can thrive in disturbed soil. So introduction, why invasive plant species? Firstly, they account for a great sum of economic loss. A 2005 Cornell study concluded that invasive species cost more than 12 billion in damages and control measures annually. However, this is not exclusive to invasive plant species, which just goes to show how much they're not looked at and how much they're ignored in that term. So according to the U.S. Forest Service, quote, invasive species have contributed to the decline of 42% of U.S. endangered and threatened species. And for 18% of U.S. endangered or threatened species, invasives are the main cause of their decline. Furthermore, invasive plant species, they degrade the wildlife habitat and cause biodiversity loss. That's mainly because they can't provide the same ecological services. But however, we need biodiversity to have clean air and good quality soil and have fresh water. Why did I specifically look at hiking trails? So the main reason is when I was looking at the research, I found that there was a lot looking at invasive species near roadsides. But I also wondered, why did we not look at hiking trails? Because in America, hiking is a really big activity that a lot of people like to do. As a matter of fact, it's the second most popular outdoor activity. So my research question is, how do hiking trails impact the spread or the distribution of non-native species and native species in Bern Preserve and Los Altos Hills? The independent variable is distance from the trail. The dependent variables that I looked at were the quantity of native and non-native plants and the quantity of species of native and non-native plants. My hypothesis was, as the distance between the trail and the location increased, the quantity of invasive species would decrease since there's less human movement to spread the species. So looking at my materials and methods. So the materials that I used were a light meter, a phone, measuring tape, and flags. So I began off by randomly picking a plot. Mostly, there was a really intense randomizing method, but I won't get into it because it would take too long. But when I reached the location, I would measure a 2 meter by 2 meter square plot. Then I would set flags at each of the 4 corners so I can actually tell where it would start and end. And then I would measure the distance from the trail. I would measure it from the bottom left flag. Then I would count the number of plants and then figure out the number of species of each plant. I would take a picture of the various plants so I can identify them later since there's no service in the area. I figured out a really random trick, but the only thing I was able to use was actually Snapchat. I went on it and then I was able to write the plot number and then the number of the quantity of the plant that I was able to find of each species. After that, I took down the latitude using a compass. The next, I measured the LUX using the light meter. However, I didn't end up using this data. After I was done, I was able to go home. I would identify the plants using apps such as Seek and iNaturalist, and also the database that I created using my own research. So the results. So this is looking at overall the quantity of native plants, native and non-native. So as we can see, the native plants, as we went further away from the trail, there was an increase in native plants and then a decrease in non-native plants. The pattern is the same here, except we're looking at the species. So let's say 30% would be 30% of for example, I would find 4 species. Half would be native and half would be non-native. It wasn't specifically looking at how much of each of them. So the discussion. There was no significance in terms of p-value. However, we did see a decrease and an increase as we've seen earlier and as I've described. Some possible reasons I didn't see significance was there was restoration work going on in the preserve for several years, which could have affected my data. Furthermore, I only did 7 plots because it just took so long to do each of them. So if I was able to do more, I'd probably be able to get better data. Another issue was that this experiment is not controlled. There were too many factors that I could not control for example, the history of the location. Something else was the accessibility to the locations that I found data from. So although they were far away from the trail, I felt like they weren't far enough, but if I would have gone any further, it would no longer be safe for me. So for future research, I would say I would look at like if somebody were to conduct the same research as me, or if I were to conduct it, I would probably say just look at more locations in different parts of the world just to get a full grasp of the concept, and also just create more plots because 7 was definitely not enough. And also just have a more experienced team to identify the plants in a more reliable way.
Great job, Farida. That's awesome.
Thank you. The best part would be probably collecting the data. As hard as it was being around all these mosquitoes and like the coyotes showing up, it was really dramatic. But I would say just being able to really just like understand where the data is coming from and be part of the collection process. I would definitely want to continue in this kind of research in the future, but I feel like now I've learned so much about what to do and what not to do, and also how to deal with challenges because definitely many came up. I've changed my the way that I look and collect the data multiple times throughout, so I feel like now I'm able to deal with that better and maybe not get frustrated or be more nervous that it won't work or will work.
Awesome, Farida. Thank you so much for sharing and for giving us a little more insight as to what it was like doing that kind of research out there. Mosquitoes are never that fun as part of field research, but
Yeah.
coyotes might be. Always kind of keeping it exciting out there. So really exciting though to hear about your research and some really interesting results that you presented on. So thanks for sharing. And Katie, thank you so much for your help with both Farida and Annette throughout this entire summer. Katie's been an excellent mentor for both of our students here that have presented so far tonight. So we appreciate all your hard work, Katie, this summer. Thanks.
Thank you.
Thank you, guys.
All right, so I'm actually lucky to introduce our next presenter, Claire Chu. Claire is now starting 11th grade at Gunn High School. And I jumped in as Claire's mentor about halfway through the summer. And what really impressed me from the moment I joined Claire and her research was the way she quickly focused her attention on the research topic that she's going to speak on tonight, and how well she worked to collect her samples in the field for this project, and also the fine level of detail she gave to the chemical and physical measurements she's made on those samples. Now, she has lots of really cool pictures that I loved looking at of all her samples at her dining room table as she worked really hard to collect these measurements over the course of the summer. And what I think is also really cool is that her research that she's going to present on follows up with what Farida just presented on, I think, very nicely. So with that, I'm going to go ahead and let Claire take it away.
Okay, can everyone see my screen?
We can, Claire. You can, yeah.
So hi, my name is Claire, and today I will be talking about my research where I investigated the effects of human activity on soil quality at Foothills Park. So I decided to research soil because it is a very important part of Earth's ecosystems. It supports plant and animal life, it regulates water flow, it cycles nutrients, and it filters out pollutants. So since soil has so many functions, I thought it would be really useful to study how its quality is affected by human activity. And here I define soil quality as its ability to carry out the functions I mentioned above. But quality can't be measured directly because it is so subjective. So I used indicators, which include chemical indicators, so pH, which influences the availability of nutrients in the soil, amount of nitrogen, which is important for plant growth and is a part of chlorophyll, which is used in photosynthesis, phosphorus, which is important in root and seed development, as well as crop maturity, and potassium, which protects plants against disease and extreme weather. I also studied a physical indicator, texture, which is percent sand, silt, and clay in the soil. It reflects how much water the soil can hold, how fast water moves through the soil, and how workable and fertile the soil is. So through all these indicators, I think they would give me a good idea of the soil quality. So my research question is, how does varying levels of human activity affect the soil quality at Foothills Park in terms of physical and chemical properties? And human activity means the amount of trampling or foot traffic that occurred in the sites. My independent variable was the locations with differing levels of human activity, and my dependent variable was the percent sand, silt, and clay, as well as the measurements of pH, nitrogen, phosphorus, and potassium. I hypothesized that human activity negatively impacts soil quality and predicted that locations with higher amounts of human activity will have lower soil quality. To test my hypothesis, I selected the 3 locations within Foothills Park with varying levels of observed human activity. Orchard Glen picnic area had the highest level, since many people have barbecues there and there's often people having parties. Chamise Trail had medium levels, since people go there to hike all the time, but it's not one of the most popular trails, so it wouldn't have as much foot traffic as the picnic area. And then the hills behind the Interpretive Center had minimum levels, since it's by the road and people don't often come out to walk there. I then chose 3 sites within each location for 9 sites total and collected soil samples at 15 centimeters deep at each site. Here's a closer look at the map of where all my sites were, as well as a picture of me in action taking soil samples. Once I collected my samples, I started with the chemical tests. Each jar was filled with half a cup of soil and 2.5 cups of water, shaken and left to settle. I then ran the tests for pH, nitrogen, phosphorus, and potassium for each of the 9 sites using the Luster Leaf Rapid Test Soil Test Kit. And then for the physical tests, I emptied and cleaned the jars, then refilled each of them with 200 grams of soil and 200 milliliters of water. I shook the jars, left them to sit for a few days, and then I measured the depth of each soil layer. Once I had all of my data, I ran ANOVA tests, and what I found was that pH did not differ among the 3 sites, with an average of 6.8, 6.7, and 6.8. And for reference, pH ranges from 0 to 14. Average phosphorus levels also did not differ among the 3 sites, with averages of 2, 2.33, and 2. Neither did the average levels of potassium, with averages of 3, 2.33, and 2.33. And for reference, the potassium and phosphorus levels range from 0 to 4. I didn't include the graph for nitrogen because most of my sites had depleted levels of nitrogen, so it wouldn't be that useful to analyze. For the physical tests, the average percent sand did differ among the 3 sites, which was pretty exciting, with averages of 87.22, 94.31, and 88.36%. But the average silt by location did not differ, with averages of 12.78, 5.69, and 9.48%. And I didn't include the graph for clay because, like with nitrogen, there wasn't that much clay in the soil, so it wouldn't be that interesting to analyze. I also plotted my soil texture data onto a soil texture triangle, which allows you to see the different, it allows you to see what type of soil your sample is. And for all three of my sites, the soil was either sand or loamy sand. So in general, my data did not support my hypothesis that human activity negatively impacts soil quality. None of my chemical tests were significant. The only significant data was the percent sand from physical tests. And sandy soil loses moisture really quickly and struggles to hold on to nutrients. So more sand could mean worse quality. And I reasoned that perhaps all this human trampling that was going on in those sites caused soil erosion, which would lead to more sand in my samples. However, the significance could also be due to external factors. For instance, at all of my sites, except for the trail sites, I took samples at 15 centimeters deep. But at the trail site, I took surface level samples just because the soil was so hard and it was like practically impossible to dig through. So that could have affected the makeup of the soil. Other external sources of error could have been that the locations just had fundamentally different environments, which would have made comparing them really difficult. I also had a very small sample size, which could have led to the high variability in my measurements and therefore resulted in no significant differences. If you go back and look at my graphs, the error bars are very large, so the variability could have been due to my small sample size. And the chemical tests were subjective, so an error in judgment could have affected my data. I've included a picture of one of the tests on the right, and as you can see, the color doesn't really match with any of the references, so it was really tough trying to choose like what the level of the chemical was. In the future, if I were to conduct a similar experiment, I would collect samples from other parks to have a larger sample size and make sure that the areas have similar environments so they're easier to compare. I would also test biological indicators. This is one of the indicators that I didn't test this time around. I only tested chemical and physical indicators. But in the future, I could look at plant height or biodiversity or the amount of organic matter at my locations. And in this experiment, I only looked at the amount of trampling as my definition of human activity, but I'm also curious how other human activities such as camping or barbecuing could affect my soil quality. So these are my works cited, and that was my presentation. Thank you for listening, and if you have any questions, feel free to type them in the chat.
All right, Claire, thanks so much for sharing. Really cool to see everything come together here for the presentation on your research. So excellent job on that. So we'll go ahead and let some questions hopefully come in. So I have a question for you. You mentioned how one of the biggest, I think, challenges was the subjective nature of looking at your chemical tests. Were there other challenges you faced out in the field? In addition to like your example of having a hard time maybe digging down in the soil, were there other issues you came across, and maybe can you tell us how you might have resolved those while you were out in the field?
I think generally my soil collecting process was pretty smooth. I just had to go there and then randomly select some sites and then just dig. So I guess the only difficulty I had while I was collecting was that some spots were really hard to dig down into. So maybe because I only picked areas that were easy to dig into, that could have affected my results as well. But besides that, I think I just had a really good time going out and doing hands-on research.
Awesome. That's what we love to hear. That's great. We have one other question coming in. So even though you did not look at biological properties, did you notice anything different while you were collecting your soil? So for example, more or less insects in some locations. So yeah, good question. You mentioned that you were maybe interested in that. Did you see anything while you were out there, even though you didn't test that?
Well, I didn't really notice much like microorganisms or like earthworms, but I did notice that the environments were different. Like the picnic area there were a lot more trees, so there's a lot of like dead leaves on the ground, and the soil was darker. Whereas on the trail, like I have some pictures where like the color of the trail is literally orange because the soil there is just different. So I guess the main thing I noticed was just like the physical environment and how they were different from each other.
Great. All right. Well, we will wrap up there for Claire. Claire, thank you so much for sharing that research. As I mentioned, it was a great experience to help you along the way as your mentor this summer. Great job all around, and really, really cool stuff that you did out there this summer. So thanks for sharing. We appreciate it.
All right, and thank you for mentoring me. I had a really good time.
You're welcome. All right, so last and certainly not least for tonight, I'm gonna introduce our final mentor. Inga's gonna come on. Hi, Inga, how are you?
Hi, I'm great. How are you doing?
I'm doing very well. Inga is joining us from Hong Kong, I believe, currently.
That's right.
Awesome. Well, we're gonna let Inga go ahead and introduce our final research for tonight. So Inga, go ahead and take it away.
Thank you so much. My name is Dr. Inga Kanti-Chirpi. I'm finishing up a postdoc here at Hong Kong U, and I will be moving to UC Berkeley this fall. I'm a coral reef ecologist, and it was my absolute pleasure to work with Meta Rapaport this summer. She is a rising senior at Los Altos High School, where she's going to continue pursuing research this coming year through their Advanced Scientific Investigation Program. And Meta, from the beginning, was clearly very passionate about corals and coral reefs, and this really shone through a project she had completed already called the Coral Reef Conservancy Project, which is a platform that tries to educate the public about coral bleaching and ways that they can help improve the situation, actions that they can take. So without any further ado, I'm going to pass it over to Meta, who will tell you more about the research she did this summer on corals and coral reefs.
Thank you. Let me get my screen shared. Okay. Hi, my name is Meta, and I am going to be talking to you about turbidity and its effect on coral abundance. Corals are an invertebrate animal that form reefs on the seafloor. They are made up of colonies held together by calcium carbonate. Most coral reefs are located in tropical areas and cover less than 0.1% of the Earth's surface, but they are one of the most biodiverse marine ecosystems in the world and are homes to over 25% of the marine wildlife. Corals have a symbiotic relationship with algae, as shown in the slide on the left picture. It's basically the circles are the algae inside of coral cells. And this algae performs photosynthesis and harnesses light in order to provide the corals with 90% of their energy. This energy is often necessary for the coral's growth and reproduction, but light varies in the ocean due to changes in turbidity. Turbidity is how clear the water is. High turbidity can be caused by small organisms called plankton. It can also be caused by sediment and silt suspended in the water. High turbidity is basically just when less light comes through the water, and I decided to research how turbidity affects coral abundance. My overall research question was does turbidity affect coral abundance? And my alternate hypothesis was that sites at higher turbidities will contain a lower coral abundance, and my null hypothesis was that coral reef abundance will not differ under different turbidities. I went to 6 sites on Big Island, Hawaii, and I used a Secchi disc, which is basically, it's a circle and it's divided into 4 parts, and it's just black and white. And the farther you hold it apart, you can tell how far the visibility is. And I used that to measure turbidity. In the diagram in the picture that I drew out, you can see that there are 2 transects, and they were each 10 meters in length and 10 meters apart from each other. And so I used a quadrat, which is in the bottom left-hand corner, to take a picture at every single meter. And I did that at every single site, and I brought that back and put them into coral point count and derived data from there, and also made visual estimates of coral cover. I then used the data received from there in RStudio to check for any statistical difference in coral abundance under different turbidities. As for my results, I was testing to see if there was a significant correlation between turbidity and coral abundance. We tested using a linear regression and found that the p-value was below this or above the expected 0.05, which means that we didn't get any significant data shown, and there wasn't a significant relationship between turbidity and coral abundance. We also performed a Welch's t-test and saw that the p-value here was also above the expected 0.05. But when we tested sites against each other, so sites 2 and 3 and 3 and 5 specifically, we found that the p-values were below the expected 0.05, which shows that there was significance there. And also just to like get across how coral point count works, because I did use that specifically for this part of my results. Points are assigned per picture that allow me to choose what point, or the points will be assigned on the picture, and I'll be able to say what they're on. And so if they're on dead coral, I would say that, and that's how I figured out how much dead coral cover there was at the sites in total. And so I used that to see if there was a significant difference between the amounts of dead coral based on turbidity at different sites. And I saw that there was a significant difference here. There was a statistically significant relationship between visibility and the percentage of dead coral cover. And turbidity could be the reason for the 83% of variation in the dead coral data. However, this should be interpreted with caution because one point is driving the entire relationship, which is site 2 on the very right. If we remove this point, then the relationship would be non-existent. So my alternate hypothesis was not supported. We did not see a relationship between coral abundance and turbidity. Using a Welch's t-test, we also checked for significant differences between specific sites and noticed that site 2 and 3 and 3 and 5 had significant differences. This showed us that there could have been other factors aside from turbidity that is causing differences in coral abundance. I noticed differences in the amount of humans at different locations, which is humans leave a huge effect on coral reefs, so this is really important. And the accessibility to the reefs were also different. There were also differences in ocean current, and this could have played a role in differences in coral abundance. When we compared dead coral cover at different turbidities, we saw that there were higher amounts of dead coral in water with higher visibility, which is not what we expected at all. However, the significant relationship once again is being driven by Site 2's data only, and at Site 2 we saw large amounts of dead coral under more visible water. It is possible that factors like disease could have caused this as well. And that is something I'd like to look into more. My results match my null hypothesis. However, we did see some significant data as mentioned earlier, so it was interesting to see how turbidity did affect factors of coral abundance, knowing that there weren't any significant effects on the overall abundance. As for why I thought I saw the results I did, there were many uncontrolled differences between sites, for example, location, human population, wave action, and more, and this could have played a role in why we saw significant differences in coral abundance at different sites. For future research, if I were to continue the project I currently worked on, I would definitely go to more sites and do more transects per sites, at least 3, so that I could get more data and get more significant results. And I wouldn't have this issue of where one point is driving all my results and I get to test this out and see if whether this driven conclusion is actually completely accurate or not. I would also like to see how the percent of dead coral cover might change if I estimated it visually. And I would also like to look into human visitation, current, and other factors and see how that may affect coral abundance. I'm also doing ASI in school, which is this competitive research program. So I will be continuing research on the coral species, and that's going to be a lot of fun for me. This is my works cited, and yeah, I overall had a lot of fun doing this, and research makes me really excited. So thank you for listening, and let me know if you have any questions.
Awesome job.
Thank you.
So I have a question for you.
Okay.
Did you see any differences between the data that you got from your visual estimates and the data that you got from coral point count? And if so, what were they?
Yeah, so when we were first putting our data into coral point count, a lot of the times the points wouldn't land on the specific corals. And so the estimate that I was taking in and what the software was giving us could have been like 2 separate things, which is why like estimating it visually is also super important because it needs to be taken into account. But coral point count gives us the possibility to get more results that we don't control because humans like to control everything. So
moving forward, would you make any changes about how you use coral point count?
I think I would apply a lot more points because I only applied 10 points per quadrat, and that didn't really, like I said, it didn't land on the corals every time, and so it might have not given me the most accurate results. But if I did like a lot more than that, and I know we were talking about this, like 100 or more, then I would probably get more significant data.
So I guess I would also love to hear what was the most challenging part of doing field work.
I think the most challenging part was actually like swimming in the ocean and having to take the picture from the same height every single time in different depths, because I don't know how to scuba dive, and so it was like constantly like going down and coming up for a breath like every 10 seconds, which was like, I think it was a really fun experience, and I also love Hawaii, so it worked out, and yeah.
Awesome. Thank you so much for sharing and giving us a little more insight as to what it was like to do that work out there in the field, and some of the things you dealt with out there. So awesome research though, Amanda. Thank you so much for sharing. It's been a real privilege, and I think a pleasure to have you doing this real cool research as part of the summer research experience. So thank you so much for sharing.
Thank you for giving me the opportunity. I had a lot of fun.
Great, and I think it was just so amazing to hear how excited you were about your research and to be out in the field. So keep that passion up, as I think one of our commenters said. Definitely keep that up.
I definitely will. Thank you.
You're welcome. And I want to go ahead and thank Inga for obviously her mentoring with Meta, but also for those of you who were able to watch last night for our presentations, Inga was not here, unfortunately, but another one of her students, Kaden, presented yesterday. So again, Inga, thank you so much for your help with those students this summer. We really appreciate all your guidance and your assistance with their projects.
Thank you for this opportunity. It's joy.
Great, thank you. All right. So well, with that, we're going to wrap up our one last fantastic night of our student presentations. As I've said the last 2 nights as well, I just want to thank all of our researchers for their hard work throughout the entire summer. We're really very proud of each of you. I think even with the collection that we showed tonight, as well as the last 2 nights, you really got a chance to see the diversity of some of the research that our students are able to do in our research experience, whether it's in the field, doing data collection with corals, or doing work with soil samples, or collecting some really cool data via online web resources, such as work that's been done with our researchers doing work with COVID. So again, a big thank you to our students for all your hard work this summer. Now, for those of you who are watching that still have a question or a comment for any of our researchers that we didn't get to, please be sure to drop those on YouTube or Facebook, and we'll make sure that the students are able to see those. And again, if you're interested as a student, or you know of a student who may be interested in joining us this September for our fall research experience, please head over to headwaterscienceinstitute.org to find more details. I also wanted to thank, of course, each of our mentors for their continued guidance for our students all summer long. This research experience would not be possible for our students without them, so a big thank you. And then finally, thanks to all of you for watching the student presentations. We hope you can all join us again for our fall presentations, so please be on the lookout for more information in the coming months. Good night, everyone.